Did you know that every cell in your body has its own skeleton? Now, when I say skeleton, pretty much most of you immediately thought of that calcified matrix we think of as bone. Your humerus, your femur, your hips, your ribs, your skull. Well, here we're not talking about that. We're talking about a protein matrix that is responsible for maintaining the shape, maintaining the ability of the cell to do work. If you look at here is what they call cytoskeleton. And think of that word, cytoskeleton. Cyto is just another fancy way of saying cell. The cell skeleton does the same thing that your calcified matrix skeleton does. It maintains the shape. Your skeleton maintains the shape of your body. Turns out, this network of proteins maintains the shape of the cell. Holds organelles in specific positions. Ditto with your skeleton. Holds your different organs. Here's where your lungs go. Here's where your spleen goes. Here's where your intestines go. It allows movement. Movement of the cytoplasm. Turns out the cytoplasm, that aqueous solution, has its own little current inside. And it also allows those vesicles, those little membrane bubbles, to be able to move around from the ER to the Golgi to the plasma membrane. Also allows for movement of the cell. Think muscle contractions. Just as your skeleton is composed of bone and cartilage, 2 components, turns out the cytoskeleton has 3. 3 different forms of these protein matrices. Each form has its own proteins that comprise it. You have the microfilaments, intermediate filaments, and then tubules. I like to make the joke, think Goldilocks and the 3 bears. Too hot, too cold, just right. Too small, too big, just right. Microfilaments are the smallest, thinnest. Microtubules, big tubes. They're the biggest. And then there's the in-between, the intermediates. Different size, different composition, different function. Previous figures, illustrations of the cells, just showed you the organelles. It didn't show you, as we see here, this meshwork of different protein filaments. Here's the intermediate filaments. Here's the microfilaments. Microtubules spanning across. Each has their different roles. The microfilaments, composed of a protein known as actin. Actin, each of these little balls you see here is a different actin protein, its quaternary structure. It then joins together with other actin subunits, and they string together and they form this chain. The chain has some rigidity to it, not much, but just enough. Because it is very simple, it turns out the microfilaments are the easiest to make, the easiest to break. Put them together, take them apart. They're very dynamic in that way. But on the flip side, Microtubules. Microtubules take a lot of proteins to put them together. They're hard to assemble, thus they're harder to disassemble. So these are more the staid components of the skeleton. Actin, put together, take apart. Put together, take apart. Tubules, put together, maintain. I like to use the illustration of a building. Everybody has watched or seen them putting together a new building. Once the foundation's there, what do they do next? They put up girders, whether they're made out of steel or wood. These girders, their job is to support, to hold up the ceiling, push out the walls. They're pushing out, pushing out, pushing out. You don't go through once the building is made and remove the girders, because then the whole building falls. Same thing here with the tubules. You put them together and their job is to support, to push out the plasma membrane, to maintain the three-dimensional shape. And because they're so structurally strong, we're going to find them in the weirdest of places. Turns out that eukaryotic cells can also have their own cilia flagella. Remember at the first lecture of this chapter, we talked about bacteria having cilia flagella. These were protrusions outside the cell. These were appendages outside the cell, not protrusions. They're appendages, things added outside the cell. Well, turns out we have these too. The cilia, small little filaments that push the plasma membrane out, still within the cell. The cells lining your bronchi, bronchioles, your trachea, have these cilia. And what these cilia do is they move in a wave-like pattern. Each cell will have hundreds of these protrusions, very short protrusions. Every time you breathe out, the cilia move in a certain pattern, a specific pattern, to pull the mucus up out of your lungs, the alveoli to the bronchioles, bronchioles to the bronchi, bronchi to the trachea, on up to your throat, and then down your esophagus to your stomach. We find these also in protozoa. Protozoa use these small little appendages. They'll be covered in thousands of them. They use them to move water around them. You move enough water, well, now you're swimming. Turns out eukaryotic cells also have a much longer protrusion. Remember here, a protrusion means that the plasma, it's still wrapped by the plasma membrane. We have a flagella. Our flagella is different than the bacterial ones. The bacteria is just a long chain of proteins that whip around behind it. Breaks off easily, replaced just as easily. Ours, not so much. Ours is still an internal component. As you see here in this picture, the flagella is actually still wrapped by the plasma membrane. Within it are long microtubules, which is kind of interesting because the cells that have flagella, the flagella can be 3 or 4 times the length of the cell, but they're still going to be at the center of them. These long microtubules that are arranged in this circular pattern of doublets. They call it the 9+2. 1, 2, 3, 4, 5, 6, 7, 8, 9 plus 2 in the center. The microtubules have proteins connecting them that move back and forth, making that long flagella beat back and forth. You've seen videos, I'm sure, of sperm. That tail coming out the back, that flagella just beating back and forth wildly. It's not spinning around like you would see with bacteria. It's just beating back and forth, kind of like if you were trying to swim with one leg and you're kicking back and forth as hard and as fast as you can. What's interesting, this 9+2 configuration, not only is it found in flagella, it's found in the cilia. The cilia of the single-cell protozoa known as Tetrahymena. It's been around hundreds and hundreds of millions of years, swimming around in puddles and streams. The 9+2 configuration is the same as you're going to find in the human sperm cell, in, well, basically all mammalian sperm cells. It's quite similar to what you're going to see in, pretty much all animals that utilize sperm in some way, or have cilia in some format. This remnant, this holdover in evolution, it's a conserved thing. It's one of the pillars supporting evolution, is that the fact that pretty much everything in the animal kingdom that has Cilia Flagella utilizes this 9+2 formation.